Welcome back, everyone, to the Connect Science webinar brought to you by the Dana Farber Harvard Cancer Center. It's a special treat to welcome a friend and colleague, Mike Heeman, from the Koch Institute for Integrated Cancer Research at MIT. Mike will be speaking on new mechanisms for old cancer drugs. Just want to remind all the attendees, if you have any questions, please feel free to post them using the Q&A tab, not the chat tab. We have an exciting list of speakers on the website that you can see indicated at the bottom of the screen. The next talk will be by Doug Green on Tuesday, October 6th, on non-canonical functions of autophagy proteins. So a few words about Mike. He did his undergraduate at Wesleyan and then graduate work with Carol Greider, followed by a very successful postdoc with Scott Lowe at Cold Spring Harbor. Came to MIT, I believe, in 2006, and has really distinguished himself as a remarkably creative scientist focused on the effects of therapy specifically within the niche and how alterations, both cell autonomous and non-autonomous, can affect the response to both old and new cancer drugs. And so it's been a pleasure, certainly for me, to work with him. And I'm really looking forward to your talk today. So Mike, please take it away.
All right, thank you, David. Thank you so much for the invitation to speak. It's really a pleasure to speak for this audience in particular, as we are consistently inspired by work coming from Dana Farber. I realize that people can't ask questions during the talk, but please ask questions afterwards. I'm really happy to talk with anybody about our work or theirs at another time. So our lab is really interested in conventional chemotherapy, the kinds of chemotherapies that are used as frontline standards of care in the clinic. And of course, much of the foundational work on chemotherapy and its early administration was done at Dana Farber. So early clinical trials and then modern implementation of chemotherapy. But a lot of this work has been really empirical and focuses on pharmacokinetics or toxicity, non-overlapping toxicity for combination regimens. And relatively little is understood about basic mechanisms of action and how tumors resist frontline chemotherapy or how they derive resistance. So I think this idea of mechanism is particularly important in the context of these therapies because mechanism allows us to guide the specific use of these therapies, in particular cancers or patients, much like we do currently with targeted therapeutics. Mechanism allows us to combine these drugs with other drugs in optimal ways. And importantly, nobody wants to give chemotherapy to patients. These are toxic drugs. So how do we actually reduce the dose of these drugs or replace them? We need to understand mechanism of action to provide other agents that provide a similar mechanism. And one thing I'd argue today is that we can actually start to understand tumor cell vulnerabilities by understanding what drugs are actually doing. So chemotherapies do unexpected things, and part of what has at least motivated us to look at these activities was work that we did in our lab and also work done in conjunction with David Weinstock's lab by Hen Losos, in which we found that cyclophosphamide, this alkylating agent that is known to induce a DNA damage response, can actually also promote an immune effect. So specifically, cyclophosphamide treatment of leukemia cells promotes a secretory response that activates macrophages. So this is a weird, unexpected effect of cyclophosphamide. It's actually promoting an immune effect in addition to what it does as an alkylating agent. So today I thought I would present three different stories related to three really boring, kind of old school standard of care chemotherapeutics, in which I think we've learned something different and perhaps unexpected about what these are doing, either intrinsically or in certain microenvironments. Work that will help us, I think, apply these more optimally in different contexts, and also to understand how we can actually promote these distinct effects in vivo. So the first work involves a study looking at cisplatin and other platinum drugs. And this is work that was done by Peter Bruno, who was a graduate student in the lab, is now a postdoctoral fellow in the Elledge lab. So cisplatin is a drug that's, as you know, is used in a wide variety of malignancies. Some 17 or 18 different cancers use cisplatin as a standard of care frontline. And in fact, it's really a terrific drug in certain cases. Testicular cancer, as you know, it can be a curative therapy. So cisplatin is thought to work by performing or forming intrastrands, so in the same strand crosslinks, or interstrand crosslinks between strands. These are thought to be the more toxic lesions between G residues in neighboring strands. And these crosslinks block the progression of DNA replication forks and also RNA polymerases, and can result then into processing into a DNA damage response. So a wide variety of cisplatin compounds are used in the clinic. And it's thought to some extent that these are interchangeable compounds. So cisplatin and carboplatin are used in the treatment of, for example, breast cancer and ovarian cancer. But other drugs like oxaliplatin are used specifically in GI cancers like colon cancer or pancreatic cancer. And so it's thought that these drugs differ in their pharmacokinetics and dynamics, but what is unclear is whether there are actually mechanistic differences between these agents. So are they used in different cancers because they have efficacy in distinct cancers? And so Pete specifically wanted to ask, so why is oxaliplatin effective in cancers where cisplatin and carboplatin are not effective? And are these therapies, this class of platinum-based chemotherapeutics, being used optimally in the clinic? And so the strategy that Pete decided to use was based on an approach that Hai Zhang, a postdoc in the lab, developed about 9 years ago. And so what Hai did is he started with a set of shRNAs that also express GFP, and he used these shRNAs individually to infect lymphoma cells. So about 50% of these lymphoma cells get infected, and then you treat them with a given chemotherapeutic. And so if an shRNA promotes resistance, then you have enrichment of GFP positive cells. If it sensitizes the therapy, then you have a decrease in GFP positive cells. And we indicate resistance as red and sensitivity in blue. So over to the right, you have this chart which shows a bunch of different shRNAs targeting genes of interest. And then on the top, we have a variety of different drugs, where again, red means resistance and blue means sensitivity. And what Hai could see, I think, reasonably easily is that this pattern of drugs, resistance and sensitivity represents perhaps a signature that defines a drug mechanism of action. So when we looked at a wider variety of drugs, so this is a subset of all the drugs that Hai and Pete and others have looked at, you actually see signatures that can really effectively classify drugs based on mechanism of action. So these patterns cluster for topoisomerase II poisons or antimitotic drugs like bimbalstin or Taxol. So we're clustering based on these signatures based on mechanism of action. So this is similar to, for example, the Broad connectivity map or other compendium approaches in yeast, but this is actually a very high resolution and very tractable signature that can separate between the action of genotoxic chemotherapies. And so an example of this is work that Pete did on a drug called CX-5461. So this is a putative Pol-1 inhibitor, but when we actually look at this signature, we can actually place it in amongst these other signatures. And here it actually tracks with topoisomerase II poisons. And so this drug is actually, I think, mischaracterized as a Pol-1 inhibitor when in fact it actually is similar to agents like topoisomerase or etoposide or daunorubicin. So it's an effective way where we can take an unknown drug and classify it based on standard or established mechanisms of action. And so Pete started working with Steve Lippard's lab, which is a platinum chemistry lab, looking at a variety of different platinum-based compounds. And so he looked at compounds including cisplatin and carboplatin, but also oxaliplatin and drugs like phenanthoplatin that the Lippard lab has made, to look to see whether they actually have common signatures or differing overall signatures. And so here's an example of a subset of these platinum drugs. So if you looked at and compared cisplatin with carboplatin, these essentially have the identical signatures. So not only do they look the same, but we can actually attach a statistical metric to this and say that essentially they cluster together in the same group. And this is to be expected because on DNA, these are essentially the same molecule. So these chlorides get cleaved off as they bind G residues here. It's identical to carboplatin when this loop gets cleaved off, and these two bonds bond with G residues. So as expected, same drug on DNA, and they have the same signature, so they represent essentially the same mechanism of action. However, he saw something very different when he looked at oxaliplatin. So oxaliplatin has a very different signature from cisplatin, and its signature is actually much more similar to phenanthroplatin. And this was interesting to us, as you might see, because phenanthroplatin actually can't cross-link DNA. It just has one of these chloride groups, and so it can only bind as a monoaddict to DNA, suggesting that oxaliplatin is similar to an agent that actually is not a DNA cross-linker. So what does oxaliplatin look like in our signature? Well, curiously, oxaliplatin looks more like drugs like actinomycin D or rapamycin, so transcription translation inhibitor. So separates distinctly from these DNA damaging agents like cisplatin or chlorambucil or carboplatin, and importantly, doesn't have this common resistance signature with P53 and CHEK2. So these are genes that are involved in the DNA damage response that essentially characterize resistance mechanisms to agents that induce DNA damage, and we don't see that in the context of cisplatin. So this is just another way of visualizing this data. This is just principal components analysis showing that cisplatin and carboplatin are over here in this territory of DNA cross-linking agents, and they separate significantly away from these transcription translation inhibitors, which include oxaliplatin and phenanthroplatin. So based on the idea that these are actually having a different mechanism of action, Pete looked at the effect of these drugs on a number of target cell lines. And these are just two examples. To the left, this is a lymphoma cell line from a mouse, and to the right, this is a human lung cancer cell line. And he looked at cell cycle responses to therapy. And what he saw is that cisplatin, or CDTP here, induces a classic S-phase G2M arrest. So this blue line here, both in the mouse cells and the human cells, whereas oxaliplatin and phenanthroplatin actually induce a really potent G1 arrest. So there are different cellular consequences in the same cell type elicited by giving these two distinct drugs. So to look at another metric for sameness or difference between these compounds, we collaborated with Junko Murai and Yves Pommière's lab at the NCI. And they had another similar signature approach here in DT40 cells. So DT40 cells are insect cells that prior to the CRISPR era were really easy to generate homologous knockouts in. And so they generated a set of knockouts for essentially all genes involved in the DNA damage response. And so in each case, for each drug, you have a central line, and everything to the left represents sensitization to a particular chemotherapeutic. So by looking at the relative patterns, we can actually, again, try to identify a signature for comparing different drugs and potentially different mechanisms of action. So I'll just focus on two areas here. This is in the area including BRCA2 and XRCC2, and below an area looking at FANC genes, and this is just sort of blown up. And what you can see is that loss of BRCA and other HR genes, as well as loss of FANC genes, really sensitizes to cisplatin and carboplatin. And this is clearly well established and is a characteristic of sensitization to platinum-based therapies. But we really don't see this particularly in the context of oxaliplatin, where in the case of, for example, FANC-D2, we really see no sensitization to oxaliplatin or phenanthroplatin. And these data, along with its similarity to phenanthroplatin, suggested to us that oxaliplatin is probably not killing via DNA cross-linked induced DNA damage, as HR defective cells are really not sensitized to these agents. So if oxaliplatin doesn't induce DNA damage, we should be able to see this. And so Pete looked at DNA damage foci, or a surrogate for these foci, looking at gamma H2AX staining. This is a modified histone that aggregates at sites of DNA damage. And what you can see is you can see a lot of these red foci in the context of cisplatin. So cisplatin is inducing an appreciable amount of DNA damage, but oxaliplatin is not. So we're really not seeing any appreciable gamma H2AX staining in these cells. And so to look at another DNA damage assay, so Pete looked at this COMET assay. So a COMET assay essentially scores the degree of DNA fragmentation following treatment. So in the case of cisplatin, you see a lot of this sort of fragmentation, so this comet emanating from treated cells. And you don't see this in the context of oxaliplatin, again suggesting that oxaliplatin at relevant therapeutic doses, so at doses that are killing these cells, is not actually inducing a significant amount of DNA damage. So what is it doing? So cisplatin induces genes like, or phosphorylation of proteins like CHEK2 and activation of P53. P53 induces proapoptotic targets like PUMA and NOXA that then antagonize BCL2, and oxaliplatin is clearly inducing apoptosis, but is not signaling through these molecules and not signaling via a DNA damage response. So just to remind you, our initial analysis suggested that oxaliplatin clusters with translation inhibitors. And I think initially we were really reluctant to believe this based on what we know about oxaliplatin, and thought that maybe our signature simply didn't have resolution to distinguish between drugs that have these different mechanisms of action. But we decided to look, I think, more generally at whether this is a possibility. And so one of the things that Pete looked at initially were genes whose expression correlates with response to both of these agents. So looked in the NCI60 cell line at genes that specifically correlated with response to these drugs. In the context of cisplatin, there's really a lack of clear correlation just by expression. So there's only one gene, Schlafen-11, that actually correlates with cisplatin response. And this is really an established cisplatin response gene. Whereas in the case of oxaliplatin, you can actually see a lot of genes whose expression correlates with oxaliplatin response, and many of these are genes that encode for the expression of ribosomal proteins, implicating this ribosomal biology perhaps in oxaliplatin response. If we just looked at GSEA analysis for categories of genes that correlate with oxaliplatin response, categories like translation elongation, translation, RNA processing, ribosome biogenesis are really at the top of this list, again suggesting that there may be a biology that correlates with ribosomal dysfunction that is induced by oxaliplatin. So to perform a genetic test to look at whether this is a ribosome-mediated stress response, we looked at a classic mediator of ribosomal stress, which is this gene RPL11. So RPL11 is a component of the ribosome that upon treatment with drugs that actually affect ribosomal biogenesis is liberated. So essentially you free up RPL11, and it's been argued that RPL11 binds to MDM2, releasing P53 to induce P53 proapoptotic targets. This doesn't quite explain the efficacy of these drugs in P53 deficient cells, but RPL11 is clearly a mediator of a broad ribosomal stress response. And so Pete reasoned that maybe if you silence RPL11, you can actually achieve resistance to ribosomal stress inducers. And so this is an example of this assay, again, performed in these lymphoma cells infected with shRNAs, where we see suppression of RPL11 really has no effect on the response to drugs like vincristine, cisplatin, and doxorubicin, but it does have an effect on drugs, on cells treated with rapamycin or actinomycin D, and it has an effect on oxaliplatin and phenanthroplatin response, again suggesting that these drugs are actually inducing a ribosomal stress, and that is mediated by these ribosomal stress inducers. Now, consistent with this, we actually see a defect in nucleolar stress, which is consistent with ribosome biogenesis defects in the presence of oxaliplatin and actinomycin, but not in the presence of cisplatin. And so specifically, if we look at the nucleolus, we can see these fibrillar caps, and these are really characteristic of nucleolar dysfunction, and we see them specifically in the context of actinomycin D and oxaliplatin treatment, but not in the presence of cisplatin. So why does any of this matter? Well, if we think about cancers that are actually treated frontline with these different agents, so colon cancers are treated predominantly with oxaliplatin, ovarian cancers are treated predominantly with cisplatin, ribosomal genes and their expression really constitutes the major difference between these tumor types. So obviously there are lots of difference between colon cancers and ovarian cancer cells, but just by expression level, if we look at the category of genes that most profoundly represents the difference between these two cancer types, they are ribosomal genes. And so perhaps this underlies the relative prevalence of treatment of these tumors with these specific drugs. So oxaliplatin in colon cancer and cisplatin in ovarian cancer. So what about tumors that actually have a mixture of these defects? So breast cancers and, ovarian cancers have predominantly HR defects. Colon cancers predominantly have APC defects. And so what we see is that if we just look at breast cancers and we look at APC expression, if you have low APC expression like colon cancers, you actually have high expression of translation machinery genes. If you actually have high APC expression, you have low expression. And if we look in general at response of these cell lines, we see high APC expressors tend to be cisplatin responsive, low APC expressors tend to be oxaliplatin responses. So again, recapitulating this clustering that we see between colon and ovarian cancer in the context of a tumor like breast cancer that actually has both kinds of alterations, so HR defects as well as APC defects. And so I think we can start thinking about these platinum drugs in really a targeted manner. So for the majority of ovarian cancers that have HR defects, obviously cisplatin is the right drug, as it is for breast cancers that have HR defects. In the context of colon cancer, most of these cancers have APC loss. And so we see them as essentially highly translational dependent or translation addicted. And so these tumors are effectively treated with oxaliplatin. But pancreatic cancers and other GI tumors are also treated with oxaliplatin. And actually, curiously, these tumors generally don't have APC defects. They tend to much more frequently have HR defects. And so we would predict at least this number of pancreatic cancers that actually have established HR defects would be more responsive to cisplatin versus oxaliplatin. And again, we can think about these tumors in a targeted fashion, so treating those breast cancers with HR defects with cisplatin and those with translation problems or APC defects with oxaliplatin. So I think the bottom line here is that chemotherapies that we think differ from one another based on toxicity or some pharmacokinetic or dynamic aspect may actually be mechanistically distinct. And I think part of the problems is that just because a drug can do something doesn't mean that it's actually doing this at relevant therapeutic doses. So oxaliplatin can cross-link DNA at high doses, but it is perhaps not cross-linked DNA that is killing cells at relevant therapeutic doses. And so I think here we need to understand resistance mechanisms at relevant therapeutic doses to really understand drug mechanism. And I think, again, importantly, understanding these mechanisms of action really may explain the empiric data that has evolved the specific treatment regimens for colon cancer and ovarian cancer. Why do we treat colon cancer with oxaliplatin versus why we treat ovarian cancer with cisplatin? And it can shed light on tumors in which there is actually significantly more heterogeneity. So the second story I wanted to talk about is work done looking at gemcitabine and resistance to gemcitabine that may be mediated by the tumor microenvironment. So as David said in the introduction, we're really interested in how tumor microenvironments promote resistance to frontline therapy. And really, our initial studies in this area were done by Luke Gilbert, who now is a lab at UCSF. And what Luke found is that chemotherapy induces DNA damage, and DNA damage obviously kills target tumor cells. So in this case, lymphoma cells. But chemotherapy doesn't only act on tumor cells, it actually also acts on the microenvironment. And in the case of lymphoma cells, we see that chemotherapy actually acts on proximal endothelial cells. And these endothelial cells, when treated, actually release pro-survival factors that counteract the action of this frontline chemotherapeutic. So this allows for the persistence essentially of minimal residual disease. So you have a subset of cells that are proximal to this protective signals. They persist following frontline chemotherapy, and they serve essentially to fuel relapse. And again, it provides an example of how a tumor microenvironment can be inherently chemoprotective. And so Simona Dahlin, a former grad student in the lab, was interested in studying this in pancreas cancer. So as most of you know, pancreas cancer is a highly heterogeneous tumor that has a great deal of stroma. This stroma includes lots of fibroblasts, immune infiltrates, endothelial cells, also includes a lot of extracellular matrix deposited proteins. And pancreas cancers are obviously a treatment intractable kind of malignancy that is thought to persist in large part due to these microenvironmental protective mechanisms. And so Simona was interested in trying to understand which factors in the pancreas microenvironment actually protect from chemotherapy. And her major focus was on treatment with gemcitabine. So gemcitabine is a frontline therapy that is used in the treatment of pancreas cancer. So it's an analog of deoxycytidine, and it's imported by the same nucleoside transporter that brings in deoxycytidine. And this will be relevant in a few slides. It's then acted upon by an intracellular kinase, deoxycytidine kinase, that actually allows this drug to persist in target tumor cells. And then it's incorporated into DNA, where it essentially arrests replication, can promote a DNA damage response, and subsequent apoptosis. This drug is generally quite effective if you actually treat tumor cell lines, but it fails to induce sustained tumor regression in vivo. So it actually can induce significant anti-tumor responses, but there is almost invariably persistent disease, and this persistent disease relapses. So Simona was really interested particularly in pancreatic stellate cells. So this is a kind of fibroblast that is abundant in the pancreas tumor microenvironment. And in fact, you see activation, so aberrant behavior of these tumor cells, both in pancreas tumors as well as in infection, so pancreatitis. And these stellate cells are thought to release cytokines and growth factors, other factors that actually promote the growth of ductal adenocarcinoma cells, so PDAC cells, and their persistence. And they've also been shown to protect tumor cells by some mechanism in vivo, although the nature of that protection, I think, has been somewhat unclear. So Simona performed a simple experiment in which she injected mice with cerulin, which actually induces pancreatitis. So it induces activation of these stellate cells. She then isolated these stellate cells and used them to condition media, so essentially secrete factors, and then put this conditioned media on top of PDAC cells, so tumor cells, in the presence of chemotherapy. So she's just looking for a factor secreted by stellate cells that promotes chemoprotection, asking, is it a soluble factor, or is it somehow direct interaction between these cell types? And in fact, she did identify a protective effect in this conditioned media. So we're looking at different dilutions of conditioned media. Here, the undiluted stuff in the dash line, in the solid line, and no conditioned media in the dash line. What you can see is there's over a log difference. So there's more than a log protection that's mediated by this soluble factor. And even when you dilute it, you actually get significant chemoprotection. This protection is actually specific to gemcitabine. So here we see treatment of gemcitabine, you have protection, but you don't with other drugs, including doxorubicin or 5-FU. And interestingly, this protective effect is also to drugs that are similar to deoxycytidine, like cytarabine and 5-azacitidine. So she obviously wanted to know, what is the protective factor in this conditioned media? So she performed a lot of complex perturbations on this, like boiling, but also proteinase K digestion, filtering, and in all cases essentially was unable to remove the factor. So it's not a protein, it's resistant to boiling, and it is smaller than 5 kilodalton. And this all suggested to us that this is actually likely a small molecule or a metabolite. And so to try to figure out what this was, we performed biochemistry, and we're not biochemists, but we performed this in collaboration with Matt Vanderheiden's lab and specifically Mark Sullivan. So they fractionated this conditioned media and identified a couple fractions here, fraction 4 and fraction 5 and 6, that contain some protective factor. And then looked by mass spec at what's actually in these fractions. And curiously enough, by far and away, the most enriched factor was deoxycytidine itself. So this shows deoxycytidine levels in these relevant fractions. So this is fraction 4, this is fraction 5 and 6, and both of these actually have considerable amounts of deoxycytidine, much more than those in other fractions or run-through fractions. So the level of deoxycytidine is actually very high. So there's about 30 micromolar of deoxycytidine, and it's present just in the presence of these activated stellate cells, but not unactivated or pre-activated stellate cells. Now, interestingly, when Matt's lab has looked at deoxycytidine levels actually in primary tumors in mice, they actually see high micromolar levels of deoxycytidine just in tumor interstitial fluid. So it's actually there in tumors themselves. We're really interested in seeing whether this is true in human tumors, although those samples, I think, have been very difficult to come by. So to see whether this, in fact, is mediating protection, Simona actually added deoxycytidine itself in the presence of gemcitabine. And what you can see here in this yellow dashed line is it promotes as a single agent significant resistance to deoxycytidine. Other deoxynucleosides actually promote some level of chemoprotection, although significantly less. So it is really an effect that is quite specific for deoxycytidine. And as I'll remind you, deoxycytidine and gemcitabine are actually imported by the same nucleoside transporter. And in fact, gemcitabine is an analog of deoxycytidine. So it's essentially the same agent. And so the model that Simona came up with, which I think makes a lot of sense, is that these stellate cells are actually secreting massive amounts of deoxycytidine, and they're really flooding out the ability of gemcitabine to do its job in areas where there's this very high concentration of deoxycytidine. As I mentioned, we think that this level actually exists in pancreas tumors. So again, these stellate cells are really secreting high levels of deoxynucleotides that are chemoprotective specifically to gemcitabine. And I think we're really amazed by this result because essentially stromal cells are specifically protecting PDAC cells from the precise agent that empirically has been shown to have the greatest efficacy in the treatment of colon cancer, which again leads us to believe that it is not random that gemcitabine is actually used in the treatment of cancer cells, that it's actually speaking to a specific tumor dependencies. And I think it's interesting to think, and it's in the area that Matt and I are very interested in, whether pancreas cells and pancreas tumor cells actually require large exogenous supplies of deoxynucleosides. I mean, I think we typically think cells can make their own basic macromolecules. But again, this data says or suggests that there's a basic dependency that the chemotherapeutic that's empirically been determined to have efficacy in this tumor is actually pointing out. So these empirically identified standards of care, I think, may actually tell us something about tumor cell vulnerabilities that we didn't realize before, that this isn't just a random drug that happens to work in this cancer based on an aspect of its pharmacokinetics or dynamics.
So Mike, I don't usually interrupt people during their talk, but I think in the spirit of the debate on Monday, I think I will. I got a question I got to ask. So, you know, there's this paper in Science from 2017 from the Weitzman and from Todd Golub at the Broad about gamma protobacteria that can eat gemcitabine. And so one possible hypothesis would be that, you know, the pancreas is colonized with these things, with these bacteria. They're eating deoxycytidine. And so actually the program is that the stellate cells make a ton of this stuff as part of a coevolution with our bacterial colonization.
Yeah, I think that's a terrific idea. I mean, I actually haven't thought about it in that term, but you know, we've thought about it in terms of what it may be doing in terms of deoxycytidine metabolism or whether there's even some sort of secretory response mediated by bacteria themselves. But that actually, I think, is a really clever idea, that they actually may be siphoning off significant amounts of this macromolecule. No, I think that's a great idea. So one of the things, we've wondered is, given the wisdom of standard of care, why is FOLFIRINOX actually used to treat pancreas cancer? As I mentioned before, we don't think oxaliplatin represents a drug that targets a cell intrinsic dependency in pancreas cancer. And so we've just looked, this is really wild speculation, at a couple pancreas cancer cell lines as well as stellate cells. And what we see, at least in very preliminary data, is that oxaliplatin 5-FU, two components of FOLFIRINOX, don't seem to combine particularly well in the treatment of these PDAC cell lines. So 5-FU seems to be doing all of the work here. Whereas in the context of stellate cells, this combination actually does seem to synergize. And I think we'd have to do a lot more work here. And this is something, again, where it'd be great to engage the broader Dana Farber community to look whether some of these agents are perhaps microenvironment targeting agents that may block the ability of stellate cells to provide a chemo-resistant paracrine signal. And again, this is consistent with the idea that David just mentioned, that other aspects of this microenvironment may have very specific effects on tumor, on drug metabolism and homeostasis in that microenvironment. Okay, so the last story I wanted to talk about was one focused on the anthracycline doxorubicin, and really an unexpected effect, for us at least, of how doxorubicin can actually induce anti-tumor immunity. So as I mentioned before, in work done by Luke Gilbert, we find that doxorubicin can actually induce a paracrine pro-survival response. And in this case, we have secretion specifically of factors like IL-6 from endothelial cells neighboring the tumor. IL-6 actually induces through the JAK-STAT pathway the expression of anti-apoptotic proteins like BCL-XL, and so the survival of these cells is due to the fact that they simply have a higher apoptotic threshold. They have higher BCL-XL in this microenvironment, and so are inherently resistant to higher doses of doxorubicin. They persist, and in this lymphoma model can fuel essentially MRD and the relapse of those tumor cells. So we're interested in looking to see whether this biology of IL-6 was true in other contexts. And so one of the models we were really interested in looking at is this BCR-ABL positive model of BALL. So this is now a well-established mouse model of BCR-ABL driven B-cell leukemia, initially generated by Richard Williams in Chuck Scher's lab. It is also an ARF deficient tumor line. So ARF is also a tumor suppressor in BALL. So this has turned out to be a really tractable model, and it's tractable because we can do transplantation experiments. You can transplant just very few cells and give rise to a disease. So it's been a great screening system. I'm not going to talk about screening today, but it's really amenable to introducing very large libraries of guide RNAs or shRNAs in vivo to look at in vivo modulators of therapeutic response. But it's also a tumor that is resistant generally, like the human disease, to lots of standard of care drugs. So these tumors can be treated with combination regimens like CVAD, in addition to BCR-ABL inhibitors like imatinib and dasatinib. So we're interested in this model to see whether IL-6 biology really has the same effect on overall response to doxorubicin. And this is work that was initiated by Eric Bent. Eric is now a radiation oncology resident at Mass General and the Brigham, and continued by Luis Milan-Barea, who's a current grad student in the lab. And so initially, they just looked at response to doxorubicin in this model. And what you can see is that upon doxorubicin treatment, you do get an extension in overall survival, but all of these mice relapse. So it's a transient effect. So they next wanted to know, what is the effect if you actually look at this biology in the absence of IL-6? And so they started essentially with IL-6 deficient mice. And they transplanted leukemia here. They waited for tumors to grow up. So these tumors grow up effectively in the absence of IL-6. So it's transplantable. IL-6 deficiency has no effect on transplantation in this context. and then treated mice with doxorubicin. And I think very surprisingly saw a significant extension in overall survival. And in fact, a large percentage of these mice are actually cured following therapy. And this is not what we saw previously in our lymphoma model. In our lymphoma model, in the absence of IL-6, you have an extension in overall survival, but all of the mice relapse. Whereas in this model, they're essentially cured in the absence of IL-6. So this is just looking at relative bioluminescence, showing that about 8 days following treatment, you had progression of tumors in wild-type recipients, but for most of these IL-6 deficient recipients, you actually have significant tumor regression, and again, many of these are actually cured. Now, so we looked at the actual direct effects of IL-6 on these tumors, as we did previously on our lymphoma model, and unlike our lymphoma model, IL-6 actually does not directly promote chemoprotection in this model. So again, as I argued before, IL-6 can signal through JAK-STAT and signal to anti-apoptotic BCL-TIL family members, but in this context, when we treat cells in the presence of IL-6, we have no difference in overall survival for in vitro treatment, cells in vitro treated with doxorubicin. So it's not directly mediated chemoprotection. And in fact, we don't see expression of the IL-6 receptor in these tumor cells. Now, IL-6 can also signal via a soluble receptor, so it's potentially possible in vivo that they're signaling through an IL-6 receptor, but at least in these cells themselves, we see essentially no expression of the IL-6 receptor. So it suggests that it's not a cell-intrinsic effect of IL-6 or the lack thereof. And so I think at the time, Eric had a really good idea that this actually may be a consequence of chemotherapy-induced immunity. So this is an idea that's actually been around for a long time, predominantly advocated by Guido Kramer. And so here basically you have treatment of a tumor cell with chemotherapy. This tumor cell is fragmented. It's taken up by dendritic cells, which then present antigens that activate the immune system. So destruction by chemotherapy can promote a sustained immune response. This is generally tested with vaccination experiments where you kill tumor cells, you inject them, and you show that you can protect mice with a live tumor challenge. And again, the Kramer lab has shown this for a number of malignancies, but it's something that I think clinically is generally not seen. So rarely do chemotherapies actually induce this sort of sustained immune response, and frequently don't elicit a sustained response at all. So there are a number of drugs that have been proposed to be these kinds of immune cell inducers or immunogenic drugs. So drugs like cisplatin and high-dose radiotherapy are thought to be more tolerogenic, so they don't induce an immune response. And the anthracyclines like doxorubicin have been shown in some contexts to induce an immune response. But again, this is generally not seen clinically. And at least in the primary response of our mice to doxorubicin, we don't see an immune effect. So if you actually perform transplant experiments into RAG-deficient mice, so immunodeficient mice, you see the same overall response as we do in wild-type recipients. So it really suggests in a normal setting, the immune system is not playing, a particularly relevant role in the clearance of these tumors. So to test the role of the immune system, Eric essentially depleted T cells in recipient mice. So he essentially depleted both CD4 and CD8 positive T cells in recipients, transplanted these tumors in, the tumors came up, and then treated them with therapy. And what he saw is that in the absence of CD4 and CD8 positive T cells, you actually have rapid relapse of these tumors following doxorubicin treatment. In the presence of immune cells, you have a sustained response. So it suggests that immune cells are actually mediating this long-term survival effect. And this is just a bioluminescence rendering of that data where, again, in the presence of immune cells, you actually have an effective remission of this disease, whereas when we deplete these immune cells, you actually have progression of disease that eventually kills the animals. So to really, I think, more strongly validate the immune effect, we performed sort of a modified immunization vaccination type experiment. In this experiment, they started with mice that are essentially cured. So these are IL-6 deficient mice that had been treated with doxorubicin and showed no tumors for a sustained period of time, and then re-injected these mice with leukemia cells. And notably, when these leukemia cells were re-injected, we never saw the re-emergence of tumor cells, whereas control mice that had never seen therapy before showed progression of disease. So in this transplant experiment, we're not reintroducing doxorubicin, we're simply reintroducing tumor cells in the mice that had previously seen doxorubicin. And these mice seem to generate a potent immune response that essentially eradicates any cells that are injected. So suggesting that this is in fact a durable immune response in this context. So what's going on? Well, I don't think we really understand what IL-6 is really doing in the immune system. One thing we know is that doxorubicin promotes a significant influx of immune cells, specifically into the bone marrow, which is the site essentially of persistent MRD in these mice. So we get a significant induction of an immune response in the presence of doxorubicin. We also see this in the absence of IL-6. We do see some difference in the immune component present in bone marrow. I think most notably we see a drop in CD4 cells, so perhaps Tregs in bone marrow in IL-6 deficient mice. And so to try to get a general sense of what may be going on, Eric actually performed global expression analysis in bone marrow, comparing wild type and IL-6 knockout mice, and identified generally mediators of inflammation as distinct in the absence of IL-6, but perhaps as you'd expect, most of these are actually down in IL-6 knockout mice. So IL-6 is a known inflammatory cytokine, and we see an absence generally of inflammation in the absence of IL-6. So what is the difference? Well, interestingly, some of the genes that are also down in this context are actually immune checkpoint transcripts. So for example, PD-1 and CTLA-4 are decreased in the absence of IL-6. This is actually at the transcript level. This is at the protein level. We actually see a drop in PD-1 expressing cells in the absence of IL-6. Now, to just try to get a general sense of whether this biology is relevant in the context of IL-6 deficiency, we performed experiments looking at PD-L1 inhibition, and sure enough, if we actually add anti-PD-L1 antibodies, we actually can significantly enhance the effect of IL-6 deficiency, or IL-6 deficiency significantly enhances the effect of the anti-PD-L1 antibody. And if we look at overall survival of these mice that are IL-6 deficient perform much better following anti-PD-L1 therapy. This can in fact achieve durable responses and even cures. So we next wanted to know, and particularly Luis wanted to know, is this a therapeutically tractable idea? What if we actually try to target the IL-6 receptor with antibodies in the context of doxorubicin treatment? And so he performed a study in which he progressively gave mice this anti-IL-6 receptor antibody and then gave them a single dose of doxorubicin. And what he saw is that in the presence of the IL-6 receptor antibody, you have a significantly enhanced response to doxorubicin. And in fact, many more of these mice show essentially long-term tumor-free survival. So there is a significant therapeutic benefit to adding this anti-IL-6 receptor. You get this durable response where mice are essentially cured in the combination of this cytotoxic chemotherapy and an IL-6 receptor antagonist. So just sort of on a side note, I'll just point out something I think is kind of interesting about these long-term survivors. So these wild type mice that are treated with doxorubicin and an anti-IL-6 receptor antibody, I'm certainly interested in engaging the community on what this may mean. And that is, if you actually take these cured mice and you now re-inject them with BALL cells and look at overall survival, these mice actually all die. And they die in the absence of any detectable tumor burden, but also in the presence of significant GI inflammation. So re-inoculation of these mice with essentially the initial agent that may promote tumor immunity or anti-tumor immunity seems to actually induce a longer-term autoimmunity in these mice. So perhaps targeting their own GI cells, which I think may be an interesting example of this kind of induced immunity following the administration of an immunogenic drug combination. So in this context, we see that IL-6 actually impairs the generation of this anti-leukemia immune response, that it promotes chemo-induced immunity which is a durable tumor immunity. And it suggests that components of standard of care regimens for BALL, I think, can be used in combination with these agents in specific ways to achieve distinct outcomes. And as I mentioned at the beginning, we've done this experiment with the Weinstock lab looking at cyclophosphamide, that perhaps cyclophosphamide functions as an immunogenic therapy in the context of perhaps rituximab treatment or R-CHOP treatment. Here, we think that we can use standard of care regimens in different ways to actually promote these specific characteristics of chemotherapeutics. And so that will end, I think, with the really big picture that I think in many cases, these frontline chemotherapies have really unexpected mechanisms of action. And we think of them in many cases as sort of interchangeable components of regimens that we optimize based on toxicity. But I think in thinking about how we actually move forward from these chemotherapeutic regimens, how do we start eliminating them or replacing them, I think understanding mechanisms and how we can actually accentuate those mechanisms is really a critical idea. So I'm going to end there and thank all of the people in my lab that did this work. I think I mentioned a lot of those in the context of the talk, but I have a great lab and a lot of terrific lab members. They're all terrific. And also terrific collaborators that I think I mentioned during the talk. So thank you again for the invitation, David, and I'd be happy to take any questions.
Mike, that was really fantastic. I just want to remind everyone to post your questions on the Q&A tab. There are some already, and I'll start. So you mentioned the signature approach for identifying drug mechanisms. Can you be a little bit more granular about when it works and when it hasn't worked in the past, and how do you think if someone has a molecule, they can make decisions about using these kinds of approaches to get at mechanism?
Yeah, so I think that one of the things I've liked about the signature is that you can ask really sort of the binary question: does it actually track next to a compound or set of compounds that we know something about, or does it not, right? So you can say by a k-nearest neighbors approach, it's in this cluster, or it's not, right? And so there are other approaches like the connectivity map, which I think are great, but it really makes a best guess, right? It says this looks like an HDAC or something, and ours sort of doesn't. And so it has a statistical component, and it also, I think, has the ability to differentiate between what we see as closely related kind of genotoxic agents. Where it doesn't work is if you have a compound that doesn't, track with the signature. And there are a lot that we've looked at where it doesn't cluster. Now, the hope is that as you build a set of compounds, you define new categories. And so the signature is tractable enough that it allows us to actually identify new categories and put things in new categories. And so, Steve Lippard's lab that works on a lot of these heavy metal compounds have been able to generate sort of new categories in that context. And it also has another, I think, kind of cool characteristic, which is that you can actually look at drug combinations and you can ask yourself, okay, you have two different signatures to begin with. What does the combination look like? And so we've done that in the past looking at things like CHOP. And so what do 4 drugs look like? And in our hands, those combinations look exactly like the average of 4 drugs. Whereas in other combinations that we see are really synergistic, it's generally one drug or the other. So you can play around because it's a simple signature and has this sort of tractable aspect to it.
That's great. So it's kind of amazing to me that no one ever looked for DNA damage during sort of the oxaliplatin drug development process. Is that really true? And I know it's been reported that the drug forms DNA adducts. Is that accurate?
Yeah, it certainly does. Absolutely. So it is clearly exerting an effect on DNA that we think is actually probably blocking Pol1 transcription. And so I won't say that it induces no DNA damage. I think it probably induces a small level of DNA damage, but I think its predominant mechanism when it's killing cells is actually through this translation effect. And I think one of the problems that occurs during drug development is you just ask this question, can this thing do this, right? And, the study we did at the beginning of the year with CX5461, this, is, if you look at the literature, it says, a first-in-class specific Pol-1 inhibitor, and that's because it was designed in its assays based on Pol-1 assays. So you specifically look at that biology, and you don't take a step back at when it's killing cells, what is it actually doing? So I think it involves having assays that look specifically at resistance mechanisms to look at why is it killing cells, when it's killing cells.
So then a question about the BCR-ABL ARF experiment. So that's a very rapidly growing model.
Yep.
Do you have any data, or have you thought about whether it's necessary to do those kinds of experiments with blockade of IL-6, say, in a context where you have a leukemia or a tumor that develops endogenously, and so the immune system has had the opportunity to see it developing and potentially develop endogenous Tregs to really understand the extent to which doxorubicin is doing this in that context?
Yeah, so that I think is a terrific question. So we haven't done that. We're currently looking at sort of germline, mice. I mean, all of our models tend to be this transplant setting, but I think that's well taken, sort of how you flip the switch, right? We've looked at different transplantable settings and see, I think varying relevance of IL-6 in different contexts. So I don't want to make the argument that this is, is any kind of magic bullet. It may actually be specific for subtypes of hematopoietic tumors where this may be in play. But I think that point is very well taken.
Yeah, there's a question also about IL-6 that's posted. Was it tocilizumab that you used to block IL-6, or was it?
This was essentially a mouse anti-IL-6 receptor antibody.
I see. So yeah, that's certainly being studied in the context of CAR-T, and I think even in COVID-19 patients.
Yeah, and I'll actually mention that there is data out there. We were not the first people that looked at the effect of IL-6 deficiency on response to checkpoint therapy. So that is really an emerging idea, and I think there are clinical trials looking at IL-6 deficiency in combination with those agents. I think, and so that was largely, I think, confirmatory in our assays. I think seeing it in the context of chemo, I think, was surprising for us.
So what are you going to study when you run out of traditional chemos, or?
Yeah, well, when you guys stop using them, I think we'll stop studying them. It's I think, as you know, it's a real challenge, and particularly in cancers like BALL, where conventional chemo has been so successful over the years. Thinking about how you actually pivot away from those therapies, I think, becomes critically important. And there are, I heard a, great talk, the other day the other day when we could go to talks months ago about, frontline therapy for neuroblastoma, and the chemo regimens get, better and better and better, and more severe, and in combinations with, bone marrow transplant. And you think that we can empirically optimize these therapies to get better anti-tumor responses, but the consequences of those treatment regimens are progressively perhaps more difficult and devastating. So how, the question is, how do we start, debulking and dissecting these regimens so that we understand who they're working for, and how to get the same action with fewer drugs or combinations with drugs that do similar things.
I don't think there's anyone who would tune into this webinar who disagrees with that statement. So at that point, I think it's really been a pleasure to host you, and thank you very much for the talk again. Everyone tune in on Tuesday for Doug Green from St. Jude's, and until then, be well. Thanks again, Mike.
Thank you. Thank you all for coming.